Hello and welcome to Medical Dialogues. I'm Anshika Mishra. Let's look at today's top medical news. Latest research debunks myths on creatine health risk. Creatine monohydrate is one of the most popular dietary supplements, widely used for boosting athletic performance and improving health outcomes. However, concerns about its safety, including potential cancer risk, kidney damage, dehydration, and stomach issues, have persisted. A recent comprehensive review published in Frontiers in Nutrition thoroughly evaluated these concerns, concluding that creatine is generally safe when used appropriately. One major worry has been whether creatine increases cancer risk due to compounds called heterocyclic amines, which form when creatine reacts with amino acids and sugar during high heat cooking. While HCAs can be mutagenic at very high levels in lab models, the amount in cooked meat and creatine supplement are significantly lower. In fact, human studies have found virtually no increase in harmful HCA production from creatine supplementation. Interestingly, some research even suggests creatine may slow tumor growth and boost cancer-fighting immune cells, although results vary by cancer type and model. Study reveals fructose consumption may silently increase inflammation levels. Fructose, the sugar found in many sweetened drinks and processed food, may do more harm than just add calories. Researchers at the University of Vienna, led by Ina Bergam, discovered that even short-term fructose consumption causes immune cells called monocytes to become more sensitive to bacterial toxins, increasing inflammation in the body. The findings, published in Redox Biology, sheds new light on how diet influences immune defenses and infection risk. The team conducted 2 randomized studies with healthy adults, comparing the immune response after drinking fructose-sweetened beverages versus glucose-sweetened one. They also isolated monocytes and ran cell culture tests to understand the biological mechanisms behind this sensitivity. Results showed that fructose, but not glucose, raised levels of toll-like receptor 2, which is TLR2, on monocytes. TLR2 plays a crucial role in detecting bacterial toxins, and its increase made immune cells far more reactive to a toxin called lipoticoic acid. Study shows maternal fatty food aroma exposure increases offspring's obesity risk. The development of obesity in children may begin before birth, influenced by factors as subtle as the smell of fatty foods experienced during pregnancy. Researchers at the Max Planck Institute for Metabolism Research discovered that exposure to the scent of fatty foods, such as bacon, in pregnant mice, even when their diet was low in fat, can prime their offspring for a higher risk of obesity and insulin resistance later in life. Their work, published in Nature Metabolism, reveals how prenatal and early postnatal environments can shape long-term metabolic health. In the study, pregnant mice were fed a healthy diet infused with the aroma of fatty foods. Despite the mother's unchanged metabolism, their offspring displayed heightened sensitivity to high-fat diets, gaining more weight and showing signs of impaired insulin signaling compared to controls. Analysis of the offspring's brain identified changes in critical regions involved in reward and hunger, notably the dopaminergic system linked to motivation and AGRP neurons controlling appetite and metabolism. These neural adaptations resemble those found in genetically obese mice, suggesting the scent exposure alone rewired brain circuits tied to energy balance. Further experiments demonstrated that fetuses and newborn mice encounter these odors both in womb and through breast milk. Remarkably, artificially stimulating the brain pathway activated by fatty food smells during early life was enough to trigger obesity in adulthood, indicating a powerful early life programming effect. Thank you for watching. Stay tuned to Medical Dialogues for more updates.
Never miss a medical update from Medical Dialogues. Like, subscribe, and press the bell icon.